Okay, we can get started. Before we jump into anything, does anyone have any questions from last week or anything that they worked on since then? Okay. Well, if there's no questions, we can jump right into it. Let me get my screen shared. Okay. And open the chat. Okay, so we've kind of gone over some of the basics of working in the command line. So running software, navigating the file system, managing files. So now we're going to get into kind of the meat of doing some bioinformatic analysis and preparing the necessary files to do so. So today we're going to be talking about writing a script. So kind of working our way into the automation process where we can write a script that's reusable for different data sets, and then we can just change file names or paths, plug in the data, and let it run. And downloading publicly available FASTQ files as well as genome reference files. So we've gone over that bit a little bit, but we'll talk about incorporating the downloading these files into a script. And just a quick, I guess, disclaimer. I think this is where it might start to move a little bit faster when we get into the scripting. So if I start going too fast or I'm not explaining something, please stop me, have me explain it again, because I don't want anyone to get lost in this process. So this week might be a little bit more challenging, but I promise that after this week, it kind of calms back down again with the tasks and challenges. Okay, and again, here's just the link to access the slides. And I'll also have the homework answers archived at this link. And as far as the recordings go, those have been uploaded to YouTube about within 3 hours after the class ends. So it's a pretty quick turnaround with that upload. Okay, so let's say that there is a data set. So you're reading a paper and they had a really interesting RNA-seq data set that you wanted to dig into some more because they answered a couple questions from the data, but you can think of other things that you want to dig into. So hypothetically, if that paper, if that data set was produced using NIH funding, those sequencing data sets are supposedly required to appear there. Doesn't always happen, but that's kind of the contract in writing, at least when people get NIH funding. So let's see. You can go to, usually these will be posted on the geodatabase. So this is the Gene Expression Omnibus. This is hosted by NCBI. And so this is where you can access all the metadata for all these data sets and find the access links that you need to either download the process data or download the raw files in case you want to start from the beginning and carry the whole processing analysis of the data set forward. So the way we do this in the command line to get the raw FASTQ files is to use the SRA toolkit. So by going through these commands, this will get you set up with the SRA toolkit on your account. So for example, I'll just walk through this real quick. So my recommendation is to have a folder called bin. So this is just kind of like a resource folder. That's the kind of normal name that it's given in Linux. And so I would download the package, the zipped package from the NCBR website. So I can do that here. But I want to do it within that bin file. So I'm just going to remove my old copy real quick so we don't run into any issues there. And then I'll just paste that command. So that should run. Oop. It looks like that recognized that as a new line, so it cut off the command. Where's the rest of that? Okay. Let me remove that bad file. Okay, so we have the zip folder, and you'll notice that, so this has that .gz link like we've talked about before, but there's additional compression. I'm using the tar utility. This is also called a tarball for whatever reason. And this, to be honest, dealing with tarball files is always a pain. I can never remember the exact flags that I need to use, but if you forgot, you just Google search, how do you uncompress a .tar.gz file? And that should lead you to using these arguments there. So I'm going to run that. It doesn't like that. There might just be a weird issue there. Nope. Okay. So I tested this earlier and it was supposed to work. So let me just double check. I left out a dash right there. And then everything else should be okay. Yeah, okay. Looks like it worked. So let's grab this. Unzip that. So you should see, just like when you unzip any directory within command line, you should see every file basically listed as it's unpacked. So then I can remove that tard file. Looks like I'm having some internet connection issues, so hopefully I don't cut out. It's making my computer very slow for some reason. Oh, come on. Sorry for all these technical issues today. Okay, there we go. So I don't need that file anymore. I can delete it. And just for my own sanity, I'm going to rename this folder to just SRA toolkit. So I'll use that move command, run SRA toolkit, and then now what I have is this folder with all the different SRA utilities. And I think those will be found within that bin folder. So it's, yeah. So here's all the different SRA applications that you can use. But we want to be able to access all of these anywhere in the system. And this doesn't, so if you'll remember when we were installing Conda, it asked if we wanted it to initialize Conda for us, so that we didn't have to manually do this ourselves. But in this case, we do have to manually do it ourselves. So I'm going to go back to my home directory. and I'm going to use this command right here. So we're going to basically export to our path. So the path is basically a collection of locations that are loaded into quick access memory, I suppose you could say, so that no matter where you're at in the system, if you run SRA toolkit, it can find that within this path. So we want to export a path variable that equals the path to that bin directory within SRA toolkits, and then this colon plus the $ path. So that's adding whatever was already in our path. It takes that old variable and appends this new path to that. And then we can just append this export command using the $! to our bashrc file. And this is that file that Condo was accessing to load its location onto. You'll notice here, this file, it begins with a period. So whenever you have a file that begins with a period, that means it's a hidden file. So if I go to my home directory and type ls, I will not see that file listed here. But if I do ls space -a, so a is for list all of those hidden files appear as well. And you can see that bashrc file is right there. Hidden files are the kind of underlying configuration of your system, and so that's why they are hidden, so that they're a little bit more difficult to access, so you don't accidentally overwrite it unless you know what you're doing. Okay, so I've already done that. I'm not going to worry about it this time. Okay, so the process of finding one of these files is, say you, one of these sequencing files, sorry, in order to use SRA Toolkit to download. So usually you should see a GSE number in the methods section. And so if I were to search that, it should be showing up. Let's see. This is GSE 54, wrong one, 825. Let's try that. Sometimes the search bar in the actual database is kind of wonky, so usually Google searching it is more efficient. So here you can see, here is the landing page for this data set. So this is a yeast data set. You can see the paper that it was linking to there. You'll have some contact information, experimental design information, and then listing of your samples. But I want to download all of those. So I'm going to go to, you'll see here, this SRA link. So this is where we're going to find the information that we need to use that SRA toolkit. So I'll click on that link. Nope, sorry, wrong link. Should have paid more attention to my slides. So the SRA run selector, this will give you a page that summarizes all of the SRA files that you can access. So you can see here some useful information. The spot length, this basically just means your average read length. So you can see here that they use 50 base pair, presumably single end sequencing. And then here you have the total number of files and the file size. But if you click on this metadata file and download that, this will give you, I need to open this in Excel. Okay. Weird, okay. file is not happy today. For some reason, it exported it as a comma-separated file, even though it's not. So I'm just going to modify that file suffix real quick. And that should load okay. There we go. Okay, so here is kind of the bulk of what we're going to need to download these files. So these are the SRR numbers. So what the SRA toolkit downloading tool needs to access this. But then you also have access to all of the metadata for these samples. So you can see these 3 samples were the biological replicates that were grown in glucose. These ones were grown in cellobiose, and so forth. So you can see a lot of other relevant, useful metadata there that may be useful as you design your analyses. Okay. So if we wanted to just download one of these files, so this one here is that first file in the set, so I can run prefetch. So this is a 2-step process that we have to use where we're basically prefetching the SRA formatted file, but then we need to convert that back into a FASTQ file. Who knows why they convert it to an SRA file? I think no one actually uses it as an SRA file. Okay. You'll probably also see this where you need to configure your SRA toolkit when you've downloaded it, but it gives you that command. So here you would type S for save, O for OK, E for exit. Oh, come on. Don't understand why. Oh, X for exit. There we go. Okay, so now that should be configured. Let's try that again. Yeah. So this is loading that file. I'm not going to carry that through. You can see how you'll get an output that eventually tells you that your download was successful. And then you can use this fastq dump command. So you specify the output directory where you want that to be. So I'm just sending that to my home directory. And then -- split file. So that's what's the file that you just prefetched that you want to convert into a FASTQ file. So this should be by default in your home directory. There's probably an NCBI folder that was created, and it should be in this file structure with the .sra file suffix at the end. And then that will convert this to a FASTQ that then you can use in your downstream processing. But what we want to do is not have to do this manually for every file. We want to basically give the command line a list of files and then do all the lifting for us. And this is nice because maybe down the road, I want to analyze a different data set. I can just modify one line that had the list of files and just substitute the new files that I want to fetch. And then I don't have to redesign the whole script or go file by file and download and dump these files into FASTQ files. So we're going to talk about a utility in the command line that is downloaded by default called Vim. So this is basically your interactive text editor in the command line. You can also use a text editor that doesn't have any formatting, such as like Notepad. And basically upload that to your folder in the supercomputer. But we're going to start with just writing it directly in the supercomputer. And you'll see here, so this ends in a .sh. So this is the file suffix that denotes a shell script file. So by running this line here, even if I don't have a file yet that's called downloadseeks.sh, this will automatically create that file for me. So this just opened that text editor. But if I had started modifying this file, saved it, and came back to it, I would just run vim download seek. So if it finds a file that's already created, it will just open that file instead of creating a new file. And so here on this slide, I've written some just helpful commands that I'll talk through as I write the script, but just so you have these for review and quick access if you get stuck. So the way that we get started right now, so if I were to start typing, you don't see anything until a while passes. But basically, you start out, sorry, my connection is slow and it makes interacting with the supercomputer slow as well. Hopefully the audio and video isn't breaking up too much though. Maybe I'm gonna close out some other windows that might be hogging bandwidth right now. Okay. So in order to enter edit mode, you need to type just I, and then you'll see down here, insert pops up at the bottom of the screen. And so that means I can start typing and editing the file. So the first step I'm going to do is write a couple of bash variables, so that this will basically allow me, if I want to reuse this script down the road, I only need to modify these couple of lines and maybe even just this line specifically. And then I don't have to remember what all the commands are. It's already designed and ready to go. So I could put in any SRR numbers and be able to download them. So with this, I have these all capitalized just to help them stand out more in the script down below, but they don't need to be capitalized. But with each of these variables, you'll give it a variable name, equals, and so if you're making a list, it needs to be between the parentheses, and each item in the list should be separated by a space. So here are the 6 SRR IDs that I want to process. I'm also going to make a variable for the output location. And so this is just output equals, and then I am just sending this to my home directory in a folder called seek files. And I think it's pretty sensitive to having spaces around these equals. You shouldn't have any spaces when you're initializing these variables, or it might be unhappy about that. The final thing I'm going to do is just that location where the prefetched files were going. So in that NCBI folder that SRA Toolkit created, I'm going to list that as a variable too. And so the first step is I'm going to create this output directory using that variable. And so after a variable is initialized in a shell script, when you want to reference that variable, you need to put that $sign beforehand. So that's just the indication to show that what's going to follow is going to be a bash variable and not just text that you should interpret literally. So this would basically make that directory right there. And then the next step is I'm going to loop through all the files and fetch the, do that prefetch step. So the step before converting it to the FASTQ file. So with this, this is going to be a for loop. And to do a for loop, so this is just going to iterate through each of the items in the list and do some action with each of those items. And so the syntax for this is, I guess, kind of makes sense with how you normally speak. So you'd say for X, or for, this can be any variable name. You just want to basically make a new variable here that you're going to reference later. You say for X in, and then we're going to reference the list up here. And in order to be able to iterate through this list, through each item, you need to have these curly brackets around the variable names, that SRR IDs. And then after the variable name, before the closing curly bracket, you need a bracket with the @ symbol and then a closing bracket. And so this is just language for bash that allows this item to be looped through and not be considered just one, basically one variable, one string. So you'd say for X in this list, for every item in the list, semicolon, do some function. And so in this case, we want to use prefetch. So that command that we talked about before, and then we're going to use that $, curly brackets, X, close curly brackets. And so this is basically going to do for each iteration through this loop, it's going to say prefetch this one, prefetch the next file, prefetch the third file. And then we're going to talk about kind of linking multiple commands into one for loop, I think on Wednesday. But since this is just one action that we want to do here, we're going to use another semicolon and type done. And so that's just going to tell the program, okay, once you've gone through all these variable names and ran this command, exit out of the loop because we're done with it. Okay, so I'm going to pause here real quick. Does anyone have any questions about any of this so far? Okay. So now we're going to create a second command that would loop through each of those variable names again and convert that SRA file to the FASTQ file. So again, the same for loop initialization here where we say for every item in the list, do something. But this time we're going to use the FASTQ dump command. And then we're going to say output directory, use that output directory. Oh, do you need to create the NCBI folder? Okay, so question about, do you need to create the NCBI folder? I'm pretty sure the SRA toolkit will automatically create that folder for you. But it's been a while, so I'd say if for some reason it doesn't create that file for you, or that folder for you, and you can't find it, let me know on Slack and we'll work through that. But it's been quite a while since I've had to deal with where that folder came from, so I don't quite remember. Another question is, what does IDs stand for in SRR IDs? Okay, so up here, this can literally be anything you want. So you're basically creating a variable here. So I could say SRR underscore list equals parentheses, and then each of these with the closing parentheses. And that would basically do the same thing. And just when I would reference this variable downstream, I would say SRR underscore list instead of SRR IDs. And that's the same with any of these variables down here. It can be whatever you want. For down here, you could say for Y, you could say for file. So you have a lot of room for variable naming when it comes to that. But you do want to avoid using a variable that's already used or already defined in the system. Because if you were to say here, if we wanted to call a variable mv equals ls. So instead of the mv function running normally in the command line, it would be running the ls command instead. And so that would overwrite the original function that was supposed to go to. Okay. So here we'll say, it's weird. Okay. I think I have a typo here. So this should be dash dash output directory, and then just that output variable here that we're referencing again with that $sign. And I think this is a typo. That should not be there. I'll have to update this slide and repost this. But then we reference the original SRA file. And so here we can use that variable that I had before where it linked to the SRA output folder. And then I can just put that slash afterwards to say, okay, now here's the next directory or file. And then I can reference the variable name for the ID here, as we did above. But it's important to remember that that prefetch command, even though we just reference the file by the ID, it's going to output a file that ends in .sra. So we need to include that here at the end. And so basically this is going to, for every loop, we'll use that same pathway variable, a different ID, and then everyone will end at .sra. And then this will create that output directory, import all the files, and convert them to FASTQ files. And then we'll be good to go. So when you're done editing a file and you want to save and quit out of the file, so you need to press escape. And so you should see that insert text disappear. And then you would do :WQ. So that stands for write and quit. And then that will return you to the command line. And then if I were to reopen that, you can see that it reopens the file as it was before. Okay. Nope. So one challenge with them is sometimes it's easy to get stuck and exit. And I think the most searched for question on programming forums is how do you exit out of them, because it is a pain sometimes. Okay, so we have that file, and so I'm just going to, so that we get some output, I'm just going to have it for x in SRR list. And we'll just print out that variable name just to prove to ourselves that it's working. And then we can save and quit this, and then any bash file is just run by typing bash space and then the file name. Oh, I think this is running my old file. Okay, so they're just printed out on each line those different IDs that we had put into that variable. So then it would process, and as it's running, it's going to output any output that you normally would have seen if you were running the command interactively in the command line. So you can see how it's progressing, but you don't have to type in the command over and over again. Okay, so I'm gonna pause again. Does anyone have any questions with creating the file, setting up the file, or executing the file? Okay. Yeah, so you should see an output like this as it cycles through each of the files. Okay, so now it's like another command or another script to download those genome FASTA files and the GTF file, the transcriptome annotation file, which will need to build the genome index that will be used to align each of the sequencing reads to that reference genome so we can know what each read, what gene each read belongs to. So if you were to just Google search, so if I want to run yeast, I would just say ensemble yeast, go to the link, and then you'll find download the GTF there. You can click there and get the link to that file to download it. And then same for the FASTA files. We go to download FASTA, DNA. And then you'll see here, so there's multiple chromosomes that we need to download. And so again, we're going to simplify this by automating the download of each of these files. And so I'll talk more about, oops, accidentally clicked on it, how we're going to do that. So again, we would create a new file. So Vim, I'm just going to call this one download refs.sh. And so you can see here, I already have this script written, but we can walk through it. So another thing that you can do when you're writing a script is include some comments. So these in a bash script are anything that starts with that pound sign. And so this will be saved to the file, but it's not actually run by the command line. So you can leave yourselves notes for what each step is doing. So you can see here, I set up this first block is for setting the variables. So I'm going to create a variable for output, which is in my home directory called reference_yeast. I have that GTF URL that I copied before. And then to get the FASTA file URL, I would just right-click on one of these files, copy that link address, and then here, this is going to give me the link for that chromosome 1 file specifically, but I don't want that whole file. I just want up until the last folder, which is here. So I'm just gonna add a space and that pound symbol to comment out the rest of that so that I remember the file name structure for each of the chromosomes that I want. And then I'm going to make another list variable that lists out each of the chromosomes here. So then again, I'm going to make a directory with that output variable that I made. I'm going to have the script. So I could have this file anywhere and run it. And this will still just automatically send me to that directory that I created so that it can just run things as if it was sitting in that directory already. And then I'm going to download the GTF URL using that curl command that we've used before. And then I'm going to use one of those looping commands for downloading each of the chromosomal FASTA files. So here I have for X and that chromosome list variable, do curl, and then I use just that FASTA path URL here, and then a slash, and then I have that file name that I just copied from the comment up here, all the way until that file name is different some way. So here you can see that the only part for each of those files is the actual chromosome number. So that's why I have up to chromosome period, and then I input the chromosome variable .fa.gz semicolon done. So after all that, I also want to unzip all these files. So they're .gz files right now. So I would do gzip space -d, so decompress. And then since all of these files I had downloaded end in .gz, I can just do *.gz, and that will just unzip all the full files in that folder ending in .gz. And then just navigate me back to my home directory. So I'm gonna save this and then run bash download refs. And then it will download each of those files. So you'll see how it's looping. This is looping probably through all the chromosomes right now. So we just let that run in the background for now. Okay, and then here's just an example of what that's going to look like. And then if we were to navigate into that directory that we made, so that one I called reference_yeast, and list the files there, you'll see the GTF file as well as the FASTA files for each of the chromosomes. Okay, so let's talk some more about some, I guess before we move on to this next topic, I'll just pause again. If anyone has any questions, we can go back and review those real quick. Okay. So when you are processing your data, you need to think through each of the steps and what are the different considerations from the experimental steps and just the underlying biology that you'll need to pay attention to when you're actually processing the data. So one really big important consideration is just genome versus RNA-seq preparation. So at face value, the raw files, you're going to get FASTQ files for both of those. They're going to look identical, but in certain downstream steps, because of the biology and how the actual reads that you're getting off the sequencer were created biologically, that will influence the tools that you need. So with genome sequencing, it's pretty, I guess, static where the reads align to positions, and that's what it is. You're not going to presumably have weird alignments or alignments due to missing chunks. I mean, there are definitely cases in genome sequencing, but generally you can just align it straight to the genome and be good. But with RNA and RNA-seq, you're dealing with processed RNA where the introns have been spliced out. And so this read right here may span 2 different exons. And so you're thinking like, okay, here's base 1, 2, 3, 4, 5, 6, 7, 8, and then here's base 20 in the genome. It will not be able to map this read with that variable intron gap in between. And so in order to be able to handle reads that span 2 different exons or 2 different regions of genomic space, the sequencing alignment tool that you'll use, you'll need to consider that as well. So for genome sequencing, you might use something like Bowtie, which just does quick alignment, but it doesn't worry about any splicing regions. But for something like RNA-seq, you probably use a tool like STAR, which in the reference genome indexing, it includes these intron spanning regions and considers that in the reference that it's building so that a read can be aligned even if it's separated by an intron somewhere. So the first step, though, that you'd want to do before getting to alignment for RNA-seq is because we ligated these adapters and these sequencing indexes to the ends of each of the reads, we need to trim those off. And we also need to remove any bad quality bases or short reads that were due to sequencing errors or just poor sequencing quality. Because if you have a read where 2/3 of it is biological sequence, but the other third is synthetic adapter sequence, that is going to prevent it from being aligned to the genome reference. And so we don't want to miss those alignments. So what I normally use is a tool called FastP. There's a lot of different tools that do the same step, such as another one is called CutAdapt. But there's some features that I really like about FastP. And so in the examples that we'll go over in this class, I'll be using that tool. The next step after you've pre-processed the reads and removed any synthetic or incorrect bases from the reads, then you would send that modified FASTQ file into a splice-aware alignment tool. And so we'll be using STAR. And so to do this, we need to generate the genome index where those splice junctions are considered. And then we need to do the actual mapping. And so you can think of like when it's building this genome index, this is very similar to how a Google search works. So basically, Google creates a dictionary of the entire internet using keywords and then can quickly say, okay, you searched this keyword and that's found on all these pages. And there's also this keyword, which is found on all these pages. And here's where those 2 lists of pages combine. And so this is probably the most relevant list of pages for you to look at. STAR is doing that with nucleotide sequence. So it's basically splitting these up into a bunch of chunks. And then it can quickly say, okay, what's your first base pair? Oh, it's an A. Okay, so now what are all the other genomic fragments that start with an A? That's kind of a simplified version of it, but that's how it's able to basically quickly search through the whole genome without basically taking, okay, here's one read, check this section in the genome. Nope, not that one, let's go move down a base pair. Not that one, move down another base pair. So that's what helps us happen in the matter of minutes to hours and not years on the computer. Okay, so as the reads that were sequenced were basically sequenced and compiled at random, a lot of tools require that these reads are sorted by position in the genome, and that just helps speed up the process downstream. And so this is where we'll be using SAM tools again to sort the alignments by genome position, and also create an index of that BAM file so that we can use interactive visualization tools that need that index to process the BAM file. And then for a generic sequencing processing pipeline, the last step you might do is quantification. So that is generating read counts from each gene. So it's saying, okay, I had these 6 reads aligned to gene A, and so gene A gets 6 read counts. And then by comparing these read counts between samples, one can get an idea of differential expression for the reads for a gene between the samples. And it's always important to run some quality control. And so we'll be using FastQC and multi-QC. And so these will make sure that the input reads that we used were good quality, that it didn't look like there were any weird sequencing biases in the data set, and so forth, so that we can be confident in the results that we do get down the line. So one thing that we can do right now is we can activate that Conda environment that we called class and download each of these files that we'll be using on Wednesday when we create our sequencing pipeline. And then we'll be able to activate this environment within the processing pipeline scripts and have all those tools available to process our samples with. So before Wednesday's class, everyone should hopefully have these tools downloaded to their CHPC environment. And then I want everyone to find a RNA-seq data set from the GEO database with 8 or fewer samples. It can be from any model organism. But what I want you to do is create a new folder, just a blank folder in the CHPC environment for the data set. And then like we went through today, download those files using SRA Toolkit. And then I also want you to, after you've downloaded all these files, copy one of the files with a new name, decompress it, and just use the head function to read the first 10 lines or 10 reads, and make sure that the FASTQ file looks normal. And then delete just that copied file that you made, that test file, but don't remove the original downloads that you had. And then as well, I forgot to include this here, but create another directory for whatever organism this RNA-seq data set was from. Create a folder in your home directory for that organism, and download the GTF file and all of the FASTA files for that organism. And then you'll be ready to basically, once you design your sequencing pipeline on Wednesday, be able to submit that job immediately to the supercomputing cluster and process that RNA-seq data set. And then we'll be using that RNA-seq data set for in the future weeks when we're using Python and R to analyze that data set that you processed and be able to perform some interesting analysis on this data set that you're interested in. So with that, we will stop there for the day. and you can answer any questions that you have or go over anything that you weren't clear on.
Hey, Jordan.
Yeah.
So I have a question regarding like the script. So when you write the script in Vim, I see like you are directly starting with the setting up the variables and all that. So I don't know, like, is it like previously I had known like when you start a script like in a bash shell or C shell, you should start with like pound forward slash bin slash bash. So is it not necessary? Like,
So it's not necessary for sure. That's just more of a stability issue. So in case the bash files, the actual like underlying program is in some weird location,
mm-hmm,
it will know where to find bash.
mm-hmm.
But in CHPC, you don't have to worry about that as much because it's kind of in that standardized position. Um,
Okay.
so if you're not running into any issues, you don't necessarily need to do that, but it's never a bad idea to do that.
Okay, so it's like not necessary, but if you are just doing like that, you can carry on. It may be a good thing or something. Okay.
Yeah. Yeah, it's not necessary until it's necessary based on the environment setup. But I think in the CHPC environment, you should be okay where that's not as necessary.
Uh-huh. Okay. Yeah. Thank you.
Yep.